A-Level Biology 9700, Paper 4, Variant 1, May, June 2025. And in this video, we will discuss question number 8 and 9. And question number 8 is, the presence of gibberellins in a plant cell leads to the expression of genes involved in stem elongation. Describe how gibberellin causes stem elongation in plants. So gibberellin is, plant growth hormone, which helps in the elongation of stems. So we have to describe how this elongation takes place. So, this gibberellin causes stem elongation by influencing gene expression through a specific signal transduction pathway. So this process primarily involves the degradation of a repressor protein, leading to the activation of genes responsible for growth. So how do this takes place? First of all, I would like to write this. In the absence of gibberellin, a family of repressor protein called DILA proteins are active and bound to the transcription factors. So the DILA protein physically represses or blocks the transcription factors from binding to the promoter region of genes. So it involves in the stem elongation. So genes for cell division, cell wall loosening factors will take place. So this keeps the stem short. So when gibberellin enters these cells, it acts as the signaling molecule. So gibberellin binds to a soluble receptor protein within the cytoplasm for the nucleus. So the gibberellin receptor complex then binds to the DILA repressor protein. So this binding marks the DILA protein for destruction by recruiting a specific E3 ubiquitin ligase complex. So DILA protein is tagged within that molecules, which signals for its degradation by the proteasome. So with the DILA repressor protein degraded and removed, the transcription factors are now free and activated. So the activated transcription factors can bind to the promoter regions of the target genes, which involved in the stem elongation. So this leads to the transcription of these genes into messenger RNA and subsequently their translation into functional proteins. So in this way, the elongation will take place. So I would like to start that how do the DILA protein, which is the repressor protein, is involved in the shortness of the stem. Then how do this gibberellin helps the plant to get elongated by the degradation of the DILA protein. So in the absence of, I would like a repressor protein DILA, DELLA, becomes active and bound to transcription factor, which blocks the elongation of stem. Gibberellin acts as the signaling molecule and binds to a soluble receptor protein within the cytoplasm and nucleus. The gibberellin receptor complex receptor complex binds to the binds to the DILA repressor protein, which marks the DILA protein for destruction. With the destruction of DILA repressor protein, the transcription factors are now free and activated. Transcription factors are free and activated, and binds to the promoter region target genes involved in stem growth. This leads to the transcription of these genes into messenger RNA and their translation into functional protein. Which includes enzyme and factors that promote cell division and cell elongation. Ultimately, cell elongates. Stem elongates. So it was all about question number 8. Moving towards the question number 9. And question number 9 is a species is classified into one of three domains. Two of these domains contain only prokaryotic species. And describe the features used to classify prokaryotic species into two different domains. So the two prokaryotic domains are, I would like to write their names, they are bacteria and second one is archaea. So these are the two domains. So uh, now we have to two prokaryotic domains describe the features used to classify prokaryotic species into two different domains. So the features are which with the help of which, first of all, that is the cell wall. And bacteria possesses peptidoglycan. And you know the archaea, they do not have peptidoglycan. So that is the reason. The first and the second one, which I would like to say the plasma membrane uh, have lipids. Bacteria have straight chain fatty acids attached to glycerol by ester linkage, whereas archaea have unique membrane lipids composed of hydrocarbons attached to glycerol by ether linkage. So this presence of ether linkage makes their membrane more resistant to heat and harsh chemical conditions. And the third, I would like to say the difference in the genetic and transcriptional machinery, which is the ribosomes. Both have 70S ribosomes and uh, RNA polymerase. Bacteria have a single simple type of RNA polymerase. Archaea have several types of RNA polymerase that are more complex and structurally resemble the RNA polymerases found in eukaryotes. So the if we talk about their habitat, so archaea often live in extreme environments such as hot springs, hot saline water, or highly acidic alkaline conditions. So bacteria are found everywhere and exhibit a vast diversity of metabolic strategies. So it was all about the, you can say, so I would like first of all the bacteria have membrane made up of peptidoglycan. Whereas yeah, don't have peptidoglycan. Instead, they have lipids composed of hydrocarbons. Secondly, both have ribosomes. Both have 70 S ribosomes. Bacteria have a single simple type of type of RNA polymerase. Whereas archaea have multiple type of RNA polymerase. So due to which they are complex, they are more complex and structurally resemble the RNA polymerases resembles with the eukaryotes. Bacteria are found everywhere, whereas archaea live in extreme environments. So it was all about this part A of the question number 9. Moving towards the second part of this question, which is the tigers are large mammals that live in many parts of the Asia. So figure 9.1 shows a tiger, Panthera tigris. Panthera tigris, and complete table 9.1 by writing the domain, kingdom and genus in which tigers are classified. So we have to complete this table. You know, the table, they have shown this table shows the classification in which the binomial nomenclature has been shown. So according to the this system of classification, they are divided into many groups and subgroups, and these, so domain is eukaryota, or eukaryotes. Kingdom is Animalia. Phylum Chordata, class Mammalia, order Carnivora, and family is Phallidae. So we have to write its genus and Panthera tigris. Panthera tigris shows that Panthera is the genus name and tigris is the species name. According to the binomial nomenclature, the name consists of two uh, groups, that is the last one, the first one genus and the second species. So Panthera. So here genus is Panthera. So the species will be Panthera. So it was all about question number 8 and 9. Please like, share, and subscribe my channel to watch more videos. Thanks for watching.